All right, so you need to be able to identify the structure and function of endospores produced by some bacteria. Not all bacteria produce endospores. We're going to name two genera, two genuses, right? Genera is actually plural for genus, that contain species that produce endospores. As I said, I'm not going to play any of the animations in class, right? But I have them here to kind of remind you guys that they're there. Sometimes, as I said earlier, they're going to go into more detail than I expect you guys to know, right? It's get the general idea of what these things are all about. We're not going to go into super specifics. So in lab, obviously, we want to be able to visualize and know if an organism has endospores. And so, as you can imagine, since we stain things to be able to see them, we have to employ special techniques to be able to stain endospores. Because of that spore coat that has keratin, that protein we have in our hair and our nails, it's a really tough protein. To be able to penetrate and get the dye into that, most of the procedures, in fact, I don't know a single one that doesn't, employ heat to get the stain to get penetrated into that spore coat. And so there are different procedures out there that use different dyes in a little bit different way of heating. But they're all going to achieve the same goal. They're going to stain the spore a particular color, and any cells are going to get stained a different color. So very much like a differential stain, right, where we can differentiate things. We can tell what's a cell, a vegetative cell, and where the spore is. So there are different methods out there, as I said. So when you're doing a particular method, you know what your results should be. Right? So this is a picture that is very similar to the method we use here in the lab at Delgado. It's called the Schaefer-Fulton method of endospore staining. And the cells get stained red, as you can see in this picture. And the endospores are a green color. Where this drawing, it's a little bit different, right? The spores are kind of a pinkish color, and the background's even stained, and the cells are not. And in this one, right, it's kind of hard to see, but again, you can see the little green spores and the red cells. So this would help us be able to visualize those structures if they're present. And as you can assume, the name endospore comes from the fact is, where is the spore? It's inside, right? Endo means within, right? So the spore actually forms within. And it's actually protecting the most precious molecule inside that cell, which is what? The DNA, right? It's stabilizing the DNA because conditions are becoming harsh. They're running out of food. They might be running out of water. The atmospheric conditions may not be right? Oxygen may be depleted, or any other changes to the different gases they may need to respire. They're sensing that something is not quite right in Denmark, and they're preparing. When they form this spore, it's called sporulization. And then eventually, over time, the cell itself around it will deteriorate. And you'll have what's referred to as free spores, spores all by themselves. Sometimes the cell itself, with the endospore still within it, is referred to as the mother cell or the sporangium. Again, all this kind of sounds like reproduction, right? It's very misleading, because at one time they probably believed it was a form of reproduction, but in fact it is not. It's a dormant period. That cell is just going into dormancy, like a bear hibernating for the winter, because there isn't much food. When these guys germinate, they basically rejuvenate. They become the original cell they once were. They're going to do that when conditions are good again. So dormant cell types. These endospores can stay dormant for hundreds of years. Just chilling, waiting for things to become good again. The bad news for us is that most forms of heat that we would employ will not disrupt this spore coat and therefore not disrupt the DNA, not destroy these endospores. Desiccation, anyone know what that means? What's desiccation mean? What gets removed when something is desiccated? Water. Water. When you dry it out. The process of actually making the endospore, it dries itself out. So if you were to try to dry it out, it's already dry to begin with. That's not going to affect it. But drying, as we'll talk about later too, right, removing water from the equation will kill most organisms, because just like us, all living organisms, every living cell out there has to have water. If water is not available, it will die. Just as we know we would die without water. Chemicals, right, as you guys will start learning about as we start doing that wiki assignment I showed you this morning. Most of the chemicals cannot disrupt that spore coat. Can't destroy those endospores. UV light doesn't penetrate far enough to damage the DNA. Can't get, again, past that spore coat. So that leaves us very few ways of being able to destroy these things. Right? There's very few chemicals out there that are effective. You have to use radiation that's much more powerful than UV light. And you have to use heat under pressure in the form of an autoclave to actually be able to destroy endospores. So the vegetative cell is produced through germination, right? And again, not a form of reproduction. The two genuses that have the most spore formers are Clostridium and Bacillus. So, anyone know a bacillus that causes a pretty serious disease that's actually an endospore former? It was sent through the mail. Anthrax, right? Bacillus anthracis, right, is a spore former. They literally sent millions upon billions of microscopic spores that people then inhaled, and those cells germinated. The bad news is that was very pathogenic bacteria. We protect ourselves on a daily basis, or even industry tries to help protect us from some of the Clostridiums. Some Clostridium tetani, right? The reason why you need to get your tetanus shot every 10 years is because that particular bacteria, its spores are prevalent in the environment, in the soil. So you could, if you have a wound that becomes anaerobic, because this is an anaerobic organism that's contaminated with spores, it could grow. And the bad news with that is the toxin it produces. Right? It stops your inhibitory motor neurons from telling your muscles to relax. Instead, they stay contracted. Hence why sometimes it's referred to as lockjaw, right? Because the patient's jaw tends to lock up the muscles that can't get them to move like they want them to. The vaccine that you get is actually against the toxin, not the bacteria. So you could still get the bacterial infection, but hopefully the toxins wouldn't get you. So what then do we have to do for a patient that might have that infection? It's a bacteria, right? Antibiotics. Antibiotics. A gram-positive bacteria. Good old penicillin will knock that sucker out. Anyone know another Clostridium? We worry about in the canning industry because it's another anaerobe. It's another spore. It's another toxin producer. Some people use this toxin cosmetically even. Botox, botulism. Botulism. Then there's Clostridium difficile. Most people that go into nursing, right, that type of care field, at one time or another, unfortunately, you may experience somebody with that type of infection. Again, it's a spore former. People ingest the spores. If there's disruption to the normal flora in the gut, that organism may take over, cause a really bad infection of the intestines. So bad it literally eats away at the intestines. And then on top of it, it smells apparently horrific. He's shaking his head. You've smelt it, huh? I have not experienced it. Thank you. Yeah, so think about it. So he said antibiotics can cause it because what did I say happens? Your normal flora, your normal bacteria in your gut, for some reason, get taken out. Well, it could have been overuse of antibiotics. Is one of the main causes that will lead to that type of infection because you kill all the good guys off. There's nobody there taking up that space in food, so when Clostridium difficile comes along, he's like, woohoo, this is all mine. And oh boy, does it take over. All right. So anyone know how they treat that? Yeah, they're going to do antibiotics, but we really got to reestablish the microbial environment in the intestines. Fecal transplants. Typically somebody in the household, right, that's been exposed to the same microbes, they're going to take a fecal sample from them and insert it into the and I'm not exactly sure how they do that. I don't want to know. But that's what they do. Right? They got to put the good bacteria back in.
I actually had a guy once that is a surgeon at Oxford Kennedy. He actually did a fecal transplant. And he told this guy, the patient's son, hey, I need you to give me some of your poop, please. Mm-hmm. And he had to give him the stuff, and they went in and actually had to insert that into the patient in some kind of way to help to do that transplant.
Right, yeah. So, I mean, you're constantly, when you poop, you get rid of a lot of your normal microbes. A lot of them stay in your intestines, but a lot come out. And that's why poop is like, oh, people are like, poop, because it can be infectious, right? Because it contains a large number of microorganisms. I mean, about 1 billion per gram, and y'all, gram is like nothing, right? That's not a lot, right? But these are good guys most of the time. But bad guys can get into the system too. And that's where, especially someone else's poop can be bad news for you if they have an infection. Did I tell you guys the kid poop story? All right. So one of my students in the past wasn't watching his 2-year-old son very well. Came into the room and the little kid had poop everywhere and had even eaten his own poop. He's like, Miss Aga, I don't understand. He did not get sick. I said, well, he just put it back in. It was his poop, right? He just put it back into his system, right? If he had put it someplace else, like for little girls, there's the whole issue of if you get it in the urethra, right, or into the vagina, you can get an infection because, again, it's bacteria. And when that bacteria gets someplace else, it can cause infection, right? But he didn't do that. He just ate it. Animals do this all the time because it still has food in it. For dogs and stuff, right, it still smells like food for some of them, right? Especially if you're giving them a very rich diet. They don't digest it all. It comes back out still a lot like food, right? But, if they're doing that, there may be some issue there, especially with animals. They might not be getting the nutrients that they need, right? So, for instance, my mom's German Shepherd, her pancreas quit producing the enzymes. So the food was going in and not getting digested and going back out the other end. So the poor dog was starving, would eat its own poop, but again, it was a vicious cycle because it was going through the system without the enzymes to digest it and actually get the nutrients. So, it can be a sign that something bad has happened. But horses, cattle, they purposely let the babies eat the mom's poop to establish their microbial environment, which is essential to their life, to establish that from eating the mom's poop. So, poop isn't all that bad, right? But it can be bad if it doesn't go where it's supposed to go, right? or if you share it with others and it's pathogenic.
I work at a daycare.
Mm-hmm.
My class is one room, we have 2 teachers and the twin. And there was a little girl, she pooped in her diaper.
Mm-hmm.
And we were going to change her, but she took her diaper off before and she smeared it all over the floor.
Mm-hmm.
And all the other kids were trying to play with it. And we was like trying to get them, no, don't touch it. But they didn't understand.
Yeah.
Don't touch it. It was everywhere. It was so bad.
Yeah, and that's the thing about little kids. So she was saying in a daycare, a little one took the poop out of the diaper, was playing with it, and the other kids wanted to play with it too, because they don't understand that that's not something you should do. We have to teach them that, right? Hygiene is something we learn, right? Which is why, little kids are little germ magnets, because they don't quite understand all of that, right? But you've got to teach them that. But yeah, certain circumstances, don't completely lose your mind over it, right? It's not completely the end of the world. But, teach. That's all we need to do. And hand washing is probably the most important thing that takes little kids a while about. So, all right, we good? All right, moving on. So, what's the structure and function of bacterial chromosomes, plasmids, and ribosomes? And what's the similarities and differences that we see in bacterial to eukaryotic ribosomes? Now remember, way back when we talked about the 3 domains, right? And what is the domain that starts with the letter A? Archaea, B, bacteria, E, eukarya. We have these 3 groupings above kingdoms because of what information did Carl Woese study? What did he study? It's on this page, y'all. There's a clue. Ribosomes. Ribosomal RNA, to be exact. Right? Ribosomes are made out of ribosomal RNA and protein. Every cell has to have ribosomes because what do ribosomes make? Protein. And so when he looked at that sequence, though, he saw these 3 distinct groups that we now call archaea, bacteria, and eukarya. So right there, we know that they differ in their RNA sequence, right? Bacteria, archaea, and eukarya. So the bacterial chromosome, unlike eukaryotes, it's not encased in membranes, right? It's not in what we call a nucleus, right? That membrane-bound organization and organelle. It's typically referred to as the nucleoid space. Typically they have a single chromosome, and what chromosomes are is not just DNA. It's protein that's associated with that DNA and even some RNA. And the protein is what helps stabilize that molecule. It's like support. So we have specialized proteins that are associated with our DNA to form our chromosome. They're called histone proteins. Right? Our DNA actually literally gets wound up around these little protein balls. And that's how we can condense our DNA down into those chromosomes that you can see. Because it's DNA, protein, and RNA. So they do the same thing. They condense their DNA with proteins and some RNA. And so you can see here, this one's been specially stained so you can see the chromosome. Typically, it's circular, it's double-stranded, right, and contains all the genetic information necessary for that cell. Now, in addition, it's very common, especially in bacteria, for them to have an additional DNA. This additional DNA, again, tends to be circular, but it's much smaller than the chromosome. Very small circular pieces of DNA. It doesn't have proteins associated with it. Not in the way in which you would see the chromosome. So notice they're 1/10 or even 10th the size of the chromosome. They're referred to as extrachromosomal. And a cell will independently replicate them, meaning they'll have multiple copies of that plasmid. Why? I don't have any earthly idea. Other than, you know, the more copies you have, the more stuff you could make from it, right? The chromosome, on the other hand, if the cell starts to replicate that, the cell is getting ready to divide. Where plasmids, they'll keep a whole bunch of copies inside their cell. Even they're not even going to divide. So that's what they mean by independently replicate. They encode characteristics that are potentially enhances survival. And so the scariest plasmids out there for us are plasmids that are known as resistance plasmids or R plasmids. These contain information, DNA, that tell that cell how to make things that destroy antibiotics. So it may be an enzyme that destroys an antibiotic, like penicillinase, or it may be it produces a different ribosome, so therefore the antibiotics that we use which bind to ribosomes can't bind to it anymore and interfere. So any information that allows them to be resistant to antibiotics, that plasmid would be referred to as a resistance plasmid. And of course, that's a benefit to the bacteria, right? It means they can survive in the presence of antibiotics. So you kind of want to keep that information. You might even want to share that information with others. Of course, for us as humans, where bacteria can cause really serious infections and even potentially kill us, that's bad news. Them having this information. So ribosomes, as we said, are what are necessary for making proteins. They're made up of two subunits, one large and one small. These come together to form the whole complex of the ribosome, which attaches to what in order to get the information to make proteins? What does it attach to? How do they know how to make a protein? RNA, right? Messenger RNA, remember that? Right? So the DNA has all the information that's encoded into messenger RNA, which is then read by the ribosome to actually make the proteins. So there's some numbers here, and this S stands for the Svinberg system of measurement. And unlike weight or length that we're used to, this combines two things. It combines almost like the weight of these molecules, but also how they move through three-dimensional space through a liquid matrix. But I always think of it, instead of thinking of it as being liquid, I think of aerodynamics, right? Notice the globular structure, but they're not smooth, right? They have little bumps and stuff on them so they can interact with the messenger RNA and the transfer RNAs and the amino acids to make the proteins. So when these molecules are by themselves, they're obviously smaller. One's larger. And you can definitely see that when you look at the size, right? 30. Ooh. I didn't even realize that. It's opposite on this slide. Right? Large should be 50. Small should be 30. How did that happen? I don't know. The total is 70, yes. So for prokaryotes, like bacteria and archaea, you've got a small piece and a large piece. And you can tell the number is small and the number is large. When they come together, the number is larger, but if you were in math class and you told your teacher that 30 plus 50 equals 70, she'd be like, no. Right? But again, this isn't just a standard one variable measurement. We're not just talking about weight. We're talking about kind of aerodynamics, how it would flow through that system. So together, obviously, it's going to be a larger number, but you can't add them together like we would traditionally do most measurements. Does that make sense to you guys? So the big question is, which numbers is Miss Erica going to make me memorize? Just the totals, right? So 70S, and you will see it in the literature, right, is always going to refer to prokaryotic ribosomes. They'll say 70S. They're talking about prokaryotes. They're talking about bacteria and archaea. Have that size designation. Eukaryotes, on the other hand, they're 40S and 60S. Do I have this animated? Nope. With a total of 80S. So what do you need to remember? 80S. Oh, you guys are waking up. And this is important too as we apply this to antimicrobials or antibiotics. Some of them literally bind to different components on the different subunits of the ribosomes and therefore interfere or stop protein synthesis. It's good news for us that our ribosomes are different from bacteria, right? Because if we took a drug like erythromycin, which targets ribosomes, if it bound to our ribosomes too, we'd be in big trouble, right? We'd be basically stopping our own cells from protein synthesis. We don't want to do that. We want to stop them from doing protein synthesis and die. So the size are different, and because of that, too, we've got different RNA sequences, and even you have that division within prokaryotes, right? Their ribosomes are the same size, but when you look down deeper into their RNA sequence, they differ. So much so that you've got two distinct groups. You've got archaea and bacteria. And then eukarya, right? We're much larger ribosomes, and again, different sequence from the prokaryotes. Still the same job, right? They all make proteins, but they do differ. And they differ enough that we can target them separately. Still with me? All right. So endocytosis and exocytosis in eukaryotes. So lysosomes play a major role in this, so that animation that goes over lysosomes is a good one to watch. So lots of eukaryotic organisms have this ability of doing endocytosis, and they even have specialized forms of endocytosis. So for instance, phagocytosis, when you take in large molecules, like for instance, we have specialized white blood cells we call phagocytes. They literally go around and take their cytoplasm, pushing their membrane, make these pseudopods, just like an amoeba eats, right? An amoeba will surround like a paramecium and put it into a vesicle. So do our white blood cells when it comes to bacteria. Right? When those guys get in, they literally go around and they gobble them up. That process is called phagocytosis. When they put them into that vesicle, we want to destroy them as well. So how do we do that? We use digestive enzymes. You don't want digestive enzymes just running free in your cell, right? Because enzymes will destroy what it is they're supposed to destroy. That's it. Like, they don't know, oh, that protein is your protein, right? They just know it's a protein that they can bind to and they can digest. So to keep ourselves from digesting ourselves, right, we take digestive enzymes and we package them in a membrane. That type of vesicle is called a lysosome. Those contain digestive enzymes. So when a cell, one of our white blood cells, ingests that, they're going to fuse a lysosome with it. Basically dump digestive enzymes onto those bacteria and kill them. And then they're going to spit it out by a process known as exocytosis, where that vesicle is going to fuse with the membrane of the cell, right, and expel the stuff outside the cell. So again, because there may be stuff there that's toxic, you don't want to leave it inside. For some of our cells, they'll even go one step further. They'll actually keep pieces of that bacteria, and not shown here, they're going to present it on their surface to be able to communicate that information to your immune system. Now, endocytosis, very large molecules, and in fact, your cells, when you're taking in a lot of fats, those will be put in a micelle, a membrane encased, and that'll actually infuse with your cell membrane and come in that way. So really large stuff can enter that same way, very large molecules. Not as large as, say, a bacteria, right? But very large molecules or substances can form a vesicle, an endosome. And again, if it needs to be digested within, we're going to do that safely within that vesicle by fusing a lysosome with it. Our cells, too, our mitochondria, were actually believed to be prokaryotic cells living all on their own, and they were engulfed by one of our ancestral eukaryotic cells way back when. Because mitochondria actually have prokaryotic ribosomes in their own DNA. But because that symbiotic relationship happened so long ago, that mitochondria cannot exist on its own. Some of the things that it needs, that information, has gone into the nucleus, is in that area, and unless the cell itself provides those things for that mitochondria, it will not continue to exist. Same holds true for chloroplasts. Right? Chloroplasts were once probably free-living photosynthetic cyanobacteria that we see today. But that symbiotic relationship, they began it, and they became so intimately involved with each other, they can't live without each other. Right? We said, okay, mitochondria, you can do all the energy processing. We'll do everything else. You handle energy processing. We'll give you the other stuff that you need. We'll maintain you. Same holds true for the chloroplasts. But because of that, when our mitochondria deteriorate, again, we're going to fuse a lysosome with it and digest it within and recycle those nutrients from that mitochondria as it ages and deteriorates. You get all your mitochondria from your mom. Right? They're in the egg. Make sense? Pretty cool stuff. All right. So prokaryotic first is eukaryotic, right? Remember, the key thing is that membrane-bound nucleus and other membrane-bound organelles. Where with prokaryotes, which are bacteria and archaea, they're still going to have things like chromosomes and ribosomes. They're just a little different, right? Yay!